All right, this is going to be part 2 of Chapter 9. We're continuing our discussion of cellular respiration and some of the biochemical pathways that are involved. And just to remind you, what I want you to walk away from this chapter with is really an understanding of, well, what are the different parts of cellular respiration, glycolysis, pyruvate oxidation, citric acid cycle, electron transport chain. How do these things fit together? What goes in, what comes out of each one of these processes? And so we're going to start here with glycolysis, which is taking place outside the mitochondria and the cytosol. All of these other steps are taking place inside the mitochondria. And if we just look at the little box up here, we can see the main thing that's happening in glycolysis is the conversion of a molecule of glucose into pyruvate. We're actually going to make two pyruvates. We're going to release a small amount of the energy that was in the glucose, and we'll store some of that in ATP, some of that in the form of reduced electron carriers. You may remember, one of the big byproducts of cellular respiration is carbon dioxide, but we actually don't make any CO2 in glycolysis. All the CO2 is coming from some of these later steps. And as we're going to see here, glycolysis involves 10 enzyme catalyzed reactions all linked together. So the product of one reaction becoming the substrate for the next. There's actually an energy investment phase in glycolysis, so we start out in the first few reactions actually having to put some energy in, use some ATP to get glycolysis started, but we're going to end up in the later reactions producing more ATP than we put in. And so in terms of housekeeping, what do I want you to know? I want you to know what goes in, which is glucose, what comes out, which is listed down here, 2 molecules of pyruvate, 2 ATP, and 2 reduced electron carriers, NADH. So for the purposes of this class, you do not have to memorize the 10 steps of glycolysis, the names of the enzymes and the intermediates. But if you are a biology or a chemistry major, you probably will have to memorize this at some point in the near future. So I did just want to walk you through it quickly so you at least have some feel for what glycolysis looks like in a little bit of detail. All right, so as I said, this is 10 reactions, and we're starting with glucose. The names of the enzymes that catalyze each step are listed in red or pink here. So here we can see the first 2 enzymes. And as I mentioned before, we start with our energy investment phase. So you'll see right off the bat, step 1, we actually have to put an ATP in. So we hydrolyze ATP, remove one of the phosphates, release some of that energy, and we use that energy that was released to attach that phosphate to our glucose. So we're left with this phosphorylated glucose molecule here. We rearrange that glucose in step 2 to make fructose 6-phosphate. And then in step 3, we spend another ATP, a second ATP, to attach a second phosphate group here. And then in the next step, we're going to split this 6-carbon sugar into 2 3-carbon pieces. One of those pieces is going to be this molecule DAP. The other will be this 3-carbon sugar G3P. And really the cell can only use in subsequent steps G3P, but so as not to waste this other 3-carbon molecule here, the cell contains an enzyme that can convert the DAP into G3P. So essentially what we're getting is 2 of these, 2 G3Ps. So just going back a slide, we're splitting this guy here into the 6-carbon sugar into 2 3-carbon G3Ps. That's basically what's happening. Now we're going to finally start to get some energy out of glycolysis. We see an oxidation reduction reaction here in the next step. So we're going to strip some electrons away from our 3-carbon sugars and give those to the electron carrier NAD+, becomes reduced into NADH. And you'll notice everything is times 2 from here on out because we have 2 of these G3Ps. We're also using a little bit of the energy from that oxidation reduction to add another phosphate to each of our little 3-carbon sugars. So we now have 2 phosphates on each one of these little sugars. In the next step, we actually produce some ATP. So we're going to steal one of the phosphates from our sugar and give it to ADP, adenosine diphosphate, to convert it into ATP. Okay. And so in this step, we make 2 ATP. So at this point, we've basically broken even, right? We spent 2 ATP in the earlier steps. Now we've produced 2. We do a rearrangement here, so we shuffle things around, shuffle that phosphate group around a little bit. We remove a water in the next step. And then in the very last reaction in glycolysis, we're going to remove the final phosphate to make 2 more ATPs. Right? So now we're ahead 2 ATP at this point. And what we're left with is 2 3-carbon pyruvate molecules. So again, what went in? Glucose went in. We get 2 pyruvates out at the end. We gain 2 ATP, and we produce 2 reduced electron carriers, 2 NADHs. So there's still a lot of energy left in the pyruvate molecules. So that pyruvate is going to get oxidized inside the mitochondria. The pyruvate will get shuttled into the inner matrix of the mitochondria. That's the very center. And it'll undergo this oxidation reaction where we will remove some electrons from pyruvate, again, give it to that electron carrier, NAD+, converting it into NADH. And we can do this twice, right, because we had two pyruvates. We'll also pull off a carbon dioxide, so this will be the first CO2 generated. And we'll use a little bit of this energy coming from this oxidation reduction to attach this chemical group called coenzyme A. And so in total, what this is going to leave us with is it will convert our 3-carbon pyruvate into this molecule called acetyl-CoA. So we produced another NADH, little CO2 as a byproduct, and we now have 2 acetyl-CoAs, which still contain some energy. And so the acetyl-CoA, let me go back slide here, sorry. The acetyl-CoA that we produce from pyruvate oxidation is now going to feed into the citric acid cycle, and we'll further extract some energy. And I'll show you the image of the citric acid cycle here in just a second. But really the key thing you need to know, what's going in. Well, acetyl-CoA is going in. You also need some of these oxidized electron carriers and GDP, but I'm not so worried about those. Main thing is acetyl-CoA is going in, and what's going to come out? Well, we're going to get 1 ATP from each acetyl-CoA that we feed in, and we're going to get a bunch of electron carriers, reduced electron carriers, so 3 NADHs, and we're going to get another electron carrier, reduced electron carrier called FADH, which is just a close relative of NADH, carries slightly less energy, but same basic idea. So we get 1 ATP, 3 NADH, 1 FADH, and we're also going to get some CO2 out, as you'll see in the cartoon here. So here's the citric acid cycle, also sometimes called the Krebs cycle. And again, you don't have to know all of these reactions or names of enzymes. I just want you to know the basic overview. But this is a true biochemical cycle, meaning as we go through these, in this case 8 reactions, we're going to end up regenerating our starting material. So as you can see, the first reaction of the citric acid cycle is combining our acetyl-CoA that's coming in with this molecule called oxaloacetate, and that combination gives us citric acid. And as we proceed through these reactions, we're basically regenerating our oxaloacetate, so we have that ready to go again if we want to feed in another acetyl-CoA down the line. And so what I want you to focus on here is, you'll see, as we go through these series of 8 reactions, there's lots of oxidation reductions going on here, right? So step 3, we're producing an NADH, right? So we're stealing electrons from isocitrate, giving them to NAD+ to make NADH, so a reduced electron carrier. So we've got one NADH being made there, another being made down here in reaction 4, another up here in reaction 8. So there's our 3 NADHs being produced. I mentioned there's another related electron carrier that will get reduced here in step 6, FADH. And down here in step 5 is where we're actually going to make our ATP. And so we're just going to get one ATP produced for every turn of this cycle. And you'll also notice we're going to get 1, 2 CO2s for each turn of the cycle as well. So I do want you to know those numbers. Citric acid cycle, for each turn, will produce 3 NADH, 1 FADH, 2 CO2, 1 ATP. Okay? And remember, we could turn that cycle twice for each glucose that we started with, right? Because we split our glucose into 2 pyruvates. Our 2 pyruvates were converted into 2 acetyl-CoAs. So we could feed 2 acetyl-CoAs in for each glucose that we started with. All right, and just kind of looking at this in overview, so this is everything that we've talked about so far. I just wanted to point out that we're basically releasing energy in lots of these small individual steps or reactions. So, here's our 10 reactions of glycolysis numbered, and we're looking at the change in free energy here in this image. And we'll see as we started glycolysis, right, we had to put some ATP in, so we see a positive change in free energy with those first few reactions. We consumed energy, and then in reactions basically 5 through 10, we released some energy. And that energy was trapped in either NADH, electron carrier, or ATP. That one reaction of pyruvate oxidation, we released more energy, right? We see another drop in delta G there, and that energy was being captured in NADH again. And then with our 8 reactions of the citric acid cycle, right, with each reaction, we see another drop in free energy. So a release of energy, again, trapping it in one of these molecules. So if we think a big picture, overall for each glucose that we started with, we would have produced, 6 carbon dioxide as a byproduct. We would have made 10 NADH reduced electron carriers, a couple of FADH, and only 4 ATP. And so if you remember, I mentioned earlier, you should be able to get up to 32 ATP for each glucose, excuse me, that you started with. So we're still leaving a lot of energy on the table here, and that's what we're going to talk about in the next section is, how do we take some of the energy that's stored in all these electron carriers and use that to make more ATP. That's basically where we're headed. So I will stop there and pick this up in the next section.